Start the recording. All right, so far we talked about the enzymes a lot, right? Proteins and their functions and stuff like that. Now we're gonna shift the gear to actually see some kind of holistic picture in terms of any kinds of biochemical reactions in the living system. So it's a metabolism, and we're gonna spend actually 4 lectures for this. So There are gonna be a lot to cover here, and basically we're using all the knowledges that we've gained so far during this course, starting with the thermodynamics, and then we talked about the macromolecules, different kinds of them like lipids, amino acids, DNA, RNAs, and stuff like that. And we're gonna see how they are flowing in terms of their energy changes and their molecular changes, their structures, and so on, through the process of metabolism, where the main purpose is to sustain the life. All right, so this is sort of a simplified version of the old maps or the flows of the molecules that are involved in metabolism. And again, this is just a simplified version. And you can see, this green portion is basically for, let me see if I can read it actually, about the sugars. So there are lots of the sugar molecules are here that are processed during the course of basically, biochemical reactions in the living organism. And this red um, region is basically for the amino acids, so the protein stuff, as well as, small or large polypeptides and so on. And you can see some, familiar macromolecules here, like a DNA, RNA stuff and things like that, which is in violet or the purple here. And also the blue regions is basically for the lipids. So there's fat and fatty acids and so on. So there are a lot of them are involved and sort of basically intermingled together to, for this metabolism processes. And we're going to mostly focus on this middle region that is yellow highlighted, which is for the process of the glucose for generating energy. So metabolism and its definition is basically a sum of all biochemical transformations in the cell. It involves breaking up the macromolecules as well as making small molecules, putting together the little pieces to make the macromolecules too. And one of the goals for metabolism is to extract energy from the energy source or the fuel, biological fuel basically, and or make the cell's own molecules that are needed for sustaining life from the precursors like the big molecules that you can uptake or intake from your diet. And there are 2 big components for the metabolism. One is catabolism and the other is anabolism. And catabolism is basically a breakdown process of complex molecules. And again, these molecules include all different kinds of biomolecules like DNA, RNA, proteins and stuff like that, etc. And this catabolism results in the energy releases. So that's an exergonic process. Again, it doesn't require energy input, so it only releases energies in most cases. And exergonic meaning that it's a spontaneous process, right? So it won't require any energy input. Again, delta G is negative. And parts of this molecules that went through the catabolism can be recaptured and further processed, or going back up to the bigger molecules. And typically starts with the old variety of different kinds of macromolecules. Again, I mean, for example, proteins have all different kinds of proteins there. I mean, even if they're built up from the only 20 amino acids, right? It just gives a lot of variabilities there, of course, because their length is different, their constituents are different, and things like that. Same thing applies to lipids or the other macromolecules like DNAs and RNAs too. But in the end, they're going to converge to the much fewer metabolites, the simple forms like simple sugar, nucleotides like adenine, things like that, or just the simple amino acids. As opposed to catabolism, anabolism is the process of synthesizing complex molecules from simple ones. So it's the opposite process from the catabolism. And it requires, of course, energy input. So that's going to be endergonic process. Again, requires energy, so it's not going to be spontaneous. It's non-spontaneous process. And it starts from the simple precursors, of course, like the simple small molecules, and then we'll build up to the some variety of the complex molecules. So both of them are important process and also happens a lot and required, of course, absolutely for the life forms. And when you think about this metabolism and catabolism and anabolism, which is like breaking down the process or the making up to the back to the macromolecule, there's a conflicting demands, right? Even for the same molecule, because there are always pathways involved for making or breaking down the same molecule in the cell. So the same molecule can be broken down, but use the same pathway to go back up to the becoming to the original state or the initial state where the it was intact, so bigger molecule. And many reactions happening in the cell are reversible, basically. So it can go back and forth and things like that. And that's a conflicting demands because sometimes you want to get certain shape or the certain state of the molecules where you want to have a smaller molecules, for example, in which case, I mean, there's a reversible reactions also can happen where it can just naturally go back to the bigger molecule state. So you have to just control it really nicely in the cell. Otherwise, it's going to just happen both ways, which is going to be pretty much like waste of energy. But the cells are designed and evolved in a way that they're working or behaving economically. So essentially, there's a no or just a little amount of waste in that cells generate in terms of energy, especially. So it's going to be either making it or breaking it if that is the same molecule. But it's not going to happen in the both ways in the cell in many cases, and that is determined by the some like needs, especially in responding to the environmental cues or some kind of like developmental cues or the demands and so on. And the cells also not wasting the energy and also do not make the unnecessary side product at all in most cases. Um, in which case they can actually discard or the waste some like little molecules or also the energy that is basically contained within that little molecules. So these cells are optimized in a way that it minimize wasting these things, but also determine the certain way to boost the biological process, biochemical processes. And again, we're going to talk about that. And it is largely, if not all, controlled and governed by thermodynamics. So we covered the thermodynamics like at least 2 times, especially at the beginning of this course, right? Quite a lot. So we're going to actually revisit some of the relevant thermodynamics here, make sure that we're on the same page. But I'm not going to dwell on it basically because it's just a review. We talked about the enzymes in the last lecture, that it basically accelerates the reaction progress to reach to the equilibrium. It's not about boosting or enhancing a certain direction of the chemical reactions or biochemical reactions. It's boosting both ways, but in the end, it'll be accelerated to reach equilibrium, right? But which way does the reaction go in the cell? Because cells do not want to waste it, right? Cells do not want to go both ways around the equilibrium. So you have to just decide one way. And again, thermodynamics is I'll take an action here where the reaction we're gonna just keep covering just the simple facts and the some knowledges that we talked about the thermodynamics here. Reactions always tend to go to the equilibrium, and at the equilibrium, the delta G will be 0, right? Basically, so there's no driving force for any kinds of spontaneous process there. And that doesn't mean that all the reactions are just in hold. It's nothing happens, dead. But it means that there's still dynamics going back and forth. But basically, there's a equilibrium, meaning that there's the same speed for or the rate for the forward reaction compared to the rate of the reverse reaction. So again, that's constantly happening, but at the same rate. And this equilibrium is set by the characteristic concentrations of reactants and products. And think about it, going back to the equilibrium and equilibrium constant. Equilibrium is basically driven by the equilibrium constant, which is defined as the ratio of the concentration of products and the concentration of reactants. And if we consider this kind of general form of the reaction here, A and B reacts to produce C and D with the different ratios here, A, B, small, little a, B, C, and D, then the KEQ, the equilibrium constant, would be basically the ratio of C and D concentration over A and B concentration. And as we talked, the coefficients of these reactions will become the exponents on the reaction rate. So it applies to the KEQ2, where it will have the C, D, and AB as an exponent here. And again, it's all about the delta G, right? So we need to basically make a relationship between the KEQ and delta G to better tell that how the reaction will go. And everything we assume that happening under the standard conditions. So we talked about chemical standard state as well as biochemical standard state. So let's quickly talk about that. The chemical standard states or the conditions is where the reaction happens under 1 atmosphere 298 Kelvin, which is 25 degrees C. And all species that are involved in biochemical reactions have to be at 1 molar. And as you know, 1 molar is a really high concentration. And again, the reaction driving force will be the delta G, which is a delta G naught in this case, which is Gibbs free energy changes under standard conditions. But when you think about some biochemical reactions, especially something happening inside the cell, everything is happening in aqueous solution, meaning that there are lots of hydrogen, cations as well as hydroxide, as well as some reactions can actually involve hydrogen cation as the reactants or the products too. So it can be a problematic because under standard conditions or standard state, you have to have hydrogen cation at 1 molar too. But again, this is a pH 0, which is a super, possible, most acidic condition. So that's never going to happen inside the cell. So that's why we had to come up with the biochemical standard state, where we have pretty much the same, the terms at the same values as 1 atmosphere or the 298 Kelvin. And all the species have to be 1 molar except for the hydrogen cation concentration, which have to be pH 7. And what would be the concentration for hydrogen cation in this case under the condition of a pH 7? Exactly, that's going to be 10 to the negative 7th, right? Remember the putting the negative logarithm to get the pH. But H2O, the water molecule concentration is set to 1 for its activity instead of 66.6 under the normal chemical standard conditions, making all the calculation easy. So here's a relationship between delta G naught, which is the delta G value at the standard state. And we can make the correlation with the delta G naught to the KEQ here using this equation. So again, this is not something completely new, right? We already discussed these equations, and you can rearrange these terms to get KEQ, which equals E minus delta G naught divided by RT. So you can relate delta G naught to KEQ here, which means that if you know KEQ, you can calculate delta G naught. And if you know delta G naught, you can get KEQ. And KEQ is the ratio of the concentration of the products and the concentration of the reactants, right? So you can actually calculate the any of those terms if you know some of the other terms. Okay, and keep in mind that this relationship, especially the delta G naught, have is located in this exponent with the negative value. So they have inverse exponent relationship, which means that the only a small changes in delta G naught will lead to the large changes in KEQ, and that's going to work in the opposite way because of this inverse exponential relationship. So that means if that delta G naught becomes negative, then KEQ will changes in the positive way. So here's an example. Here, we already talked about this, but in the table, just a small changes in delta G naught, almost like small changes, like 5 or 6 changes here, will lead to the order of like 10, basically here, 10 times differences in KEQ. Okay, so any questions so far? It's just a review again. But it's important to understand how metabolic process happens and how we can understand in terms of enzymes that are involved for driving any kinds of directions and determining forward or reverse reactions based on these values, the thermodynamic terms. So again, here we just talked about the delta G naught at the standard state, but delta G, the actual can be measured. That delta G value, the free energy under a normal condition or the physiological condition, can be different from delta G naught. I mean, temperature is different, and the concentration of the reactants are different. So we can get the delta G, calculate the delta G based on this equation again, where delta G equals delta G naught plus RT times Ln, and this is the ratio of the concentration of the reactants and the product and the concentration of the reactants. And that means even if delta G naught is positive, so delta G naught can be positive, which means that the reaction is not spontaneous at standard state, which is 1 atmosphere and 25 degrees C and so on, everything's at 1 molar. It can still happen spontaneously when delta G is negative, right? And when delta G naught is positive, this term, RT, Ln, and the ratio of these concentrations have to be negative. And that should be negative enough and big enough to get this delta G value negative, right? So that means R, again, this term has to be negative, but R is the gas constant, which is a positive value, and it's a pretty big value. And T is a temperature in Kelvin, so it's going to be, in most cases, positive value. And that means this Ln and ratio of the concentrations should be negative. And to get the negative value from the Ln, the natural log function, you have to have this value inside the log function smaller than 1, right? So it has to be smaller than 1. And that means the concentration of reactants should be larger than concentration of products. If that's the case, you're going to have a negative value for this Ln function. Then you're going to get the negative delta G if this term is much bigger than this in terms of their absolute values. And there are cases where these products, the C and D, can be cleared or removed from the other reaction by the other reactions, where other reactions use these C and D for their reactants. In this case, they're going to be kept consumed, right? In that case, the product concentration can stay low, really low. So again, you're going to have a really low value, which is smaller than 1, for this ratio of the concentrations to keep the delta G negative. So in that case, this reaction will still move forward and keep moving forward as long as it, this C and D are keep clearing, keep, removed by the other reaction. Okay, so let's talk about this removal of the products for driving the forward reaction in more details. So and even endergonic reaction, an endergonic reaction is again is a non-spontaneous reaction, right? So it requires energy input. So it's not going to happen by itself, but still can be driven by coupling with the exergonic reaction. So let's consider these two reactions here. A becomes B and B becomes C. And if you put it together, then A goes to B and B goes to C. So this B product is going to be canceled out and will work as a sort of an intermediate molecule. So the total reaction would be A to C, right? And when you consider the different delta G values here, under the standard state, that's going to be delta G naught. So A to B, the total delta G naught value would be the delta G naught value for A to B reaction plus the delta G naught value for the B to C reaction. So 1 and 2 reactions together. And that means that even if delta G1 naught is positive, if delta G2 naught is largely negative, then you can actually get the delta negative value for the delta G naught total for the total reaction too. So again, let's think that this A to B reaction is endergonic, which means that the delta G1 naught is a positive value. But if the B to C reaction is strongly exergonic, meaning that it has the highly negative delta G value, delta G naught value, then if you put it together, delta G naught total will be negative. So it can still pull the reaction to the forward direction. And again, here the B is used as a common intermediate. And that really happens a lot. That's a general way to drive this these kind of endergonic reactions in the cell to couple it with the highly exergonic process to drive it in the certain way. An example of this coupling of the endergonic process to the exergonic process in the cell is a phosphorylation of glucose, which is catalyzed by the hexokinase. So here's a reaction. Glucose that gets the inorganic phosphate, or PI, to get the glucose 6-phosphate and the water molecule. And delta G naught value for this reaction is positive, 13.8 kilojoule per mole. So it's not going to happen spontaneously. It'll require energy input there, right? But it can be coupled to the ATP hydrolysis using the common intermediates. And ATP hydrolysis process is written here, where ATP combines with the water molecule, and it's a hydrolysis. So it gets water molecule and can be broken down using that. So it's going to become ADP, which loses phosphate here. And that's G naught value is negative 30.5 kilojoule. So it's negative, highly negative, and highly exergonic process. So if you combine these two together, you can just cancel out the same molecules on the different side. So PI canceled out, and H2O2 to get the glucose plus ATP becomes glucose 6-phosphate and the ADP. And delta G naught for total reaction will be basically the sum of these two delta G naught values, which is still negative. So overall reaction would be exergonic. So in these two reactions, what are the common intermediates? Is it glucose or a phosphate or glucose 6-phosphate or water molecule? Exactly. So there are two common intermediates. that are involved here, which are inorganic phosphate and the water molecule, which are just used in the middle, and then eventually will disappear, right? So it's same as A to B, B to C, and then you see A to C, and you don't see B there. Okay? All right. So for regulating this kind of process, whether the reaction goes forward or the reverse, or how you're gonna accelerate this certain processes, especially for making the intermediates to boost the certain reaction of the direction of the reaction and things like that. Enzymes have a critical role there because it can basically boost that reactions, even if it cannot tell the equilibrium state, it cannot change it. It can at least boost the speed of the reaction at that state where it still does not meet the equilibrium yet. And because these enzymes are important for the metabolic pathway and their regulations, regulating enzymes are also really important. And it is especially important for understanding the mechanisms of metabolism. And the regulation of enzymes is basically regulating or modulating the flow of metabolites through the metabolic pathway. And again, many reactions are reversible using this equilibrium, basically. But this regulated pathway should move to the just one direction, at least predominantly, depending on its need. So and typically it's not just a simple reaction, A becomes B, right? But there are lots of intermediates involved there. So there are a lot of places where enzymes can play a critical role for driving certain reactions. And also there are a lot of places or the spaces where enzymes can be regulated for modulating the speed or the overall process of metabolism. So when you look at this, consider this reaction, just a general reaction that A can change to B, and B becomes C, and C becomes D, and it can go back and forth and things like that. There are going to be two regulation mechanisms. One would be directly changing the concentration of each players or the reactants here or the products here. For example, if you get rid of A here, then because of the equilibrium that has to met at certain point, B is going to go back to A, right? And that can pull the C goes back to B, and also D goes back to C. Same thing applies to the getting rid of this B, in which case A will quickly goes to the B, but C and D will pull down backward to the becoming B, something like that. So that's one regulation mechanism, and it's called a substrate limited reaction, where substrate plays a key role there, limiting the reaction rate. And the second possible regulation method would be modulating this reaction itself that are involved in the A to B or B to C or C to D. And they are typically boosted by the enzymes or the catalysts, right? So it's going to be modulating and changing the enzymes, its activity or some other features of them. And that's called enzyme limited reaction, because enzyme plays the key role here, again, limiting the rate of the reaction or the direction of the reaction. So let's keep talking about the regulations that are happening through the metabolic pathway. We just talked about enzyme-limited reactions, and it's not just the enzyme-limited reaction, it can be substrate-limited reaction too, but they can work as rate-limiting steps in the pathway. In the enzyme sections, we talked that the slowest reaction of the overall reaction will be basically the rate limiting steps, because it's the slowest one, so it's going to work as a bottleneck, right? Same thing applies to the metabolic pathways too, where this enzyme limited reaction, which is the slowest reaction, would be the rate limiting steps. But these reactions are typically highly exergonic. So whenever somehow some enzymes are, let's say that regulated there negatively, but per need, when it's activated a certain amount, it has to go the reaction, right? It has to work in the reaction and just to um, accelerate the reaction in the desirable way as early, as fast as possible. So that reaction has to be exergonic. So as soon as it meets the enzyme, it should just boost and kick in that reaction. So it can be used as a target of regulation. So it's like you're just stepping on the brake there, but stepping on your gas at the same time. So whenever you release your brake, you're gonna just move right away. Um, and again, this slow is the step, rate limiting step, cannot be, determines the sort of the whole reaction speed rate. So the whole pathway cannot be faster than its slow is the step. There are additional solutions or, basically ways to regulate these metabolic pathways. There are two methods here that I listed. And the first one is compartmentalization of forward and reverse pathways. That means forward, pathway or the reaction happens at one place, but the reverse pathway can happen in the other way. So you can independently regulate those pathways, which is really advantageous, especially when you want to just regulate one of those reactions, right? And an example would be the fatty acid molecule regulation, where its synthesis happens in the cytoplasm, but it's oxidation, which is for the breakdown. And we're going to talk about oxidation a little later in this lecture, happens in the mitochondria. And the second method to the regulate this metabolic pathway will be the using non-identical forward and reverse pathways. So of course, I mean, it will share the same pathway going back and forth around the equilibrium, but also you can invent the other way to come back to the initial molecule there, where you can use that pathway too to further regulate the speed or the equilibrium there. Okay. And certain enzymes are key regulatory points in the metabolic pathways, such as committed steps. So some enzymes can actually determine that this is the checkpoint, and if it actually passed that checkpoint, then there's no way to go back to the previous state. So it happens a lot in the cell cycle regulations, where you committed to go to a next phase of the cell cycle, then you don't want to go back. This is the completed state. So in which case, the enzymes will just push forward of the reaction. Another example is a branch points, where it actually make a decision there, right? So once it decide to go certain route or the pathway, and then it will go through that pathway and cannot actually go back and change the other pathway. Happens a lot during the metabolism, especially catabolism. All right. There are, and when you think about the regulations of enzymes or metabolic pathways, it's typically inhibition rather than acceleration or the promotion or enhancement, because it's an easier way to just stop it at a certain point until something is ready, like you have enough materials or the reactants there and so on. So the living systems use two largely different type of inhibitions here. The first one, and it's of course applies to the metabolic pathways, but it also can be applied to any kinds of biochemical reactions. The first one is a feedback inhibition. Let's imagine that there's a precursor, and that is processed to X molecule, and then it'll go through the different intermediates to become Y, and that's what the cell wants. But the accumulation of Y can give inhibits the conversion of X to A intermediate, where it sets the negative feedback loop like that. In that case, Y will eventually drop because the product itself inhibits the production of the Y molecule. But when this Y is enough dropped and down and pretty scarce, then now this pathway can be reactivated to synthesize Y even further. And this negative feedback loop is widely used, especially for maintaining certain state in terms of the certain chemicals or I'm sorry, certain molecule concentrations or the pH levels and things like that in the living system. Of course, living systems want to be in the under desirable conditions as much as possible, right? So it's just a balancing that ratio of the X and Y molecules here. And this process is called homeostasis. And an example would be the threonine dehydrogenase, where it uses a threonine to generate the isoleucine. And that will have that nice balance where it has to have the maintain the pretty much consistent ratio of the threonine and isoleucine. And the other inhibition type is the product inhibition. And an example would be this glucose becoming to the glucose 6-phosphate again. And there's ATP and ADP is involved. And we also talked about the hexokinase, but it's an enzyme that accelerates this reaction. And interestingly, this hexokinase enzyme is going to be inhibited by its product glucose 6-phosphate. So glucose 6-phosphate can be bound to the hexokinase, not in the active site, because glucose as well as glucose 6-phosphate would be, at least transiently bound to the active site of hexokinase, right, for its process. But it's a different site other than the active site of hexokinase where glucose 6-phosphate can bind, and then it will actually induce the conformational change, especially on the active site, to the make it more like inactive form, and it's called allosteric or allosteric inhibition, where this inhibition or the regulation happens outside of the active site. And this is a really highly exergonic process. So you definitely stop it, right? Because it's glucose, and you're basically just making into the glucose 6-phosphate and to go through the energy production pathway. And you don't want to have the energy always, right? You want to keep your energy source as it is when you have enough energy already. So this product inhibition works really nicely here, where it has a lot of glucose 6-phosphate, then just stop it. Hexokinase will stop there until there's enough glucose 6-phosphate that is already processed further for the energy generation. Okay, so we keep talking about the regulations of the enzymes that are involved in metabolic pathways. And this is a different type of regulation for the enzymatic reaction activities. It is using the spatial organization, and there are largely 3 different types of spatial organization of enzymes through the metabolic pathway. The first one is separate individual enzymes, where there are different enzymes are involved for getting the final product. But there are just a separate individual soluble proteins, so they're going to exist randomly in a cytoplasm or some kind of other places inside some kind of organelle there, like ER or mitochondria and so on. And let's imagine that there are 3 enzymes that are involved for this particular reaction to get the final product. There's a initial initial reactants that has to flow and diffuse to the enzyme A, and then it'll make the first intermediate molecule, right? And that has to flow and diffuse to the enzyme B to make the second intermediate, and that second intermediate also has to just diffuse out to get to the enzyme C to finally get the product molecule there. So it's seemingly kind of inefficient way to do it, but it's widely used types of regulations in the living system, depending on the what kind of molecules that it is processing. Second type of spatial organization is a multi-enzyme complex. So it can make either this kind of blob, kind of complex, or it can make this kind of nice, like array of the enzymes where it can work as a sort of a conveyor belt. So the advantage of having this complex is that doesn't have to wait until the certain intermediates comes to the other enzymes like B or C um, by diffusion, but it can create the substrate channel basically, again, working as a sort of a conveyor belt where the molecule stays inside the complex of the enzymes and it will process it in a much faster fashion. And the third type is the membrane-bound enzyme system. And example would be the respiratory chain or the electron transport chain, which is in the mitochondria. And that's for generating energies. And it is particularly efficient because instead of talking about the 3D space, now you can just handle the 2D space along the membrane, right? So it's a better way to, probably more efficient way to organize the enzymes and also recruits their substrates there. But also it has a limitation where everything has to be on the membrane and also have to be specialized for certain organelles and so on. But the diffusion is minimal. Okay, so far we talked about a lot of different kinds of regulations on enzymes in the different aspects. And this is another aspect which is really important and widely used in the living system for the regulating enzymes again. And that is again for the regulating and modulating the metabolic pathway. There are two regulations for enzymes themselves. The first one is enzymes availability, and you can just simply imagine that availability, the abundance, right? So it's a subject of molecular biology and molecular genetics where they modulate the rate of synthesis or degradation of the enzymes availability by changing their mRNA levels or protein levels. And mRNA levels basically is determined by a transcription rate. Protein level is regulated by the translation rate, right? And also degradation can be regulated by some transcriptional or translational regulations, as well as post-transcriptional or post-translational regulations like a phosphorylation, any kinds of things like ubiquitination and so on. Many things can be involved basically for degradation as well as the synthesis of the mRNA and protein for the enzymes. The second way is the changing the enzyme's activity. And there are mainly 5 different kinds of ways to change or modulate the enzyme's activity. The first one's allosteric enzymes. And we already talked about this allosteric, where there's some small molecules that is binding to the allosteric site, which is often different from the active site, to change the activity of the enzymes by leading the conformational change in many cases. And these effectors can be substrate itself, in which case it's called homotropic allosteric enzyme, or it can be a different small molecules, different from the substrate, in which case it's called the heterotropic allosteric enzymes. And another way to do it is by the reversible covalent modifications like a phosphorylation or ubiquitination. And many different kinds of sugar decorations, basically smorylation or myristylation and so on. And another way is to reversible binding of other proteins, and these proteins are typically controlling the enzymes for their activities. And also there's irreversible covalent modifications, especially proteolytic cleavages, where the protein or the enzyme is cut in half, or two pieces or multiple pieces, in which case it's almost impossible to put them back together, right? So it's going to be irreversible modification. And also by the changes of the localization of an enzyme too. So we're going to look at all these different kinds of methods to regulate the enzyme's activity. I know that's a lot of information, but these are just quite straightforward, and just based on the facts. So hopefully you don't get confused too much here. Um, if you have any questions, just raise your hand anytime. The first method to regulate the enzyme's activity is by allostery. And allos means other, and steros means shape. So it's other shape, basically. And it's a way to fine-tune the enzyme's activity and making this enzyme into the other shape, basically. So again, about conformational change. It induces those changes for changing, especially the shape of the active site, and that leads to the changes in the activity of the enzymes. And that is due to the modulator here, either allosteric activator or allosteric inhibitor. And again, these binds to the sites other than active sites. And what it does simply is to induce the changes in active site to either induce or inhibit the interaction with the substrates. And then typically it actually changed the conformational leads to the conformational change of all the subunits once one of subunits in the complex is changed. So an example of that is a hemoglobin that we already talked about, where it has the cooperative conformational change. Once one of those four subunits is changed its form from T, the tense form, to the relaxed form, for binding the oxygen. So it's going to have a higher affinity to the oxygen molecule, right? In that case, it will induce the conformational change of other subunits within the hemoglobin to lead to the all of them just binding oxygens more easily. So again, it's all about the cooperativity, and allostery plays a critical role for this cooperativity. Because all the subunits are attached to each other, and you can just consider one subunit as an allosteric activator for the other subunit. So in this case, of course, they're going to have two different subunits or the sites, right? One for the regulation, the other for the catalysis, the active site. So they typically have separate regulatory and catalytic subunits, and can be either activators and inhibitors in terms of the allosteric modulators that they're binding. If the allosteric site is the same as active site, again, we talked about that. If the substrate works as an allosteric modulator too, then it'll be called homotropic. If those molecules, the modulators, are different from the substrates, then it'll be called heterotropic enzymes. And these allosteric enzymes have a really unusual kinetic behavior when you look at this substrate saturation curve, or where the velocity is plotted against the changing concentration of the substrates. And this is the normal hyperbolic substrate saturation curve for the any kinds of common substrate, as opposed to when the allostery is in play, then it'll have this sigmoidal curve. And it's, again, we've already seen it when we discuss hemoglobins. And that is basically the reason, because it changed the curve when the substrate concentration changes, like from the more flattened curve to the more steep curve, is because it changes its mode. So when the substrate is low, especially for the hemoglobin, it'll have a low affinity to the oxygen or any kinds of substrates. So it'll behave something like the um, a reaction, biochemical reaction with inhibitor, basically, where you have a certain curve here. And then once the concentration of the substrate increases, then it'll change the mode from this inhibition mode to the more active mode, where it'll have a more flattened Lineweaver-Burke plot, where the Km value will change. So somewhere I actually have written here, where the curve is the ratio of Km and Vmax and Km, right? So it'll change the Km value, but it will not change the Vmax. And Km is basically the ratio of the enzyme ES complex, which is a in transition state, and the separate enzyme and substrate, right? So it changes the affinity of the enzyme to the substrate to the enzyme. So that means it'll easily form the ES complex. So it will change the curve Km, which will lead to the changes of the slope here. But the Vmax does not change because this one, the allosteric only affects the affinity, not the rate of the reaction or the efficiency of the reaction. So you're going to end it up having this sigmoidal curve for both saturation curve as well as Lineweaver-Burke plot too. Question? It definitely can. But so the question was that the question was that these allosteric effectors mostly affect the Km, but it can also change whether it can also change the Vmax too. Yes, it is. It is a possibility, depending on the how it is affecting. If it is affecting only the affinity, then it'll only change Km. If it affects the actual rate, efficiency, then it can also change the Vmax. So that's true. And this changes in slope in the Lineweaver-Burke plot reminds us some type of inhibition, right? So in the last lecture, we talked about competitive inhibition, uncompetitive inhibition, and non-competitive inhibition, or mixed inhibition, right? Which one do you think it's in play in this case for the allosteric? Is it competitive inhibition? Does it similar to competitive inhibition or uncompetitive or non-competitive? If you have reviewed, last lecture, because it only changed the curve, the slope of the line, but it does not change the y-intercept, right? Which is Vmax. It only changed the Km in this case, which is competitive inhibition. So allosteric works as the opposite way of the competitive inhibition. Question? Vmax, you mean? So imagine that. I mean, mixed. What is it? The uncompetitive inhibition, where we just actually let's just put this aside, and let's go back to the previous lecture. And when you imagine the second type of inhibition, which is uncompetitive inhibition, the inhibitor can bind to both ES complex as well as just the free E form too, right? Where it changes both Vmax as well as the Km too. So you can take it in the opposite way here. So it's not about the inhibition, but it's more like the activation. But you can take it as the recovery from the uncompetitive inhibition. So if this allosteric works for the both active site as well as the allosteric site, then it'll change both Km and Vmax. That make sense? Exactly. Exactly. Okay. All right. So for the this allosteric, where it changes the conformation of the enzyme, especially their subunits, there are two different models to explain how it works. The first one is Monod-Wyman-Changier model. It's also called the symmetry model, where it's just assumed that everything happens simultaneously. So it has both a T state, and T is the tenth state, again, for the hemoglobin as well as many other allosteric enzymes. And it can change to the R state, relaxed state, but that can happen all together for their subunits. And another model is Koshland model, where there must be a sequential changes. One, there's a change of conformational change of one subunit, and that can lead to or induce the conformational change of other subunit too. So both are correct basically. So the many different kinds of enzymatic systems in the living organism uses both strategies here, but just one of them in most cases. And that depends typically on the how many subunits you have in many cases, and the size of the subunits. The second method to regulate the enzymatic activity. So we're talking about different methods or the strategies to regulate the activity of the enzymes, right? Specifically activity of enzymes, not the availability. The second one is the reversible covalent modifications, and it's actually quite much simpler than the allosteric. So it's similar to the on-off switch or the high or low function switch or activity switch. And the typical groups that are used for covalent modifications are listed here, like inorganic phosphate or some nucleotides like the AMP or UMP, or ADP with the ribose or methyl group. And they're attached to the different amino acids in the enzymes. And as you can see, most of them are basically polar or acidic or basic. Tyrosine is widely used here. And this one has the hydroxyl group, which is reactive, and just because of the oxygen molecule there, so you can recruit the different types of these functional groups for the modification. Question. Good question. So the question was that should I memorize this? Should I be able to match which groups attached to which amino acids? I did not think about the exam questions yet, so I can't really give you the definite answer for this. Probably not. But we'll see. I mean, if you need to memorize it, I'll definitely let you know. So, and classical example of this reversible covalent modification is a glycogen phosphorylase. So you can have the two different forms. One's unphosphorylated form, and it's called glycogen phosphorylase B form, and it's relatively inactive. Or it can be phosphorylated form, which is called glycogen phosphorylase A form, and it's more active. And this B form can be active too, that we're going to talk about in the next slide. But when you look at the structure of this glycogen phosphorylase, it has dimer. Basically, it's a dimer, so it has two equivalent subunits there, and the active site is basically a little dip here that is exposed into the outward, and it also has allosteric effector site. And what would that mean? It has an allosteric effector site. What does that mean? And we're talking about reversible covalent modification. Does that mean anything? Exactly. So since it has the allosteric effector site, it can actually work, as an allosteric effector or allosteric enzyme too. Good question. It's a good catch, actually. So didn't want to explain this too much yet, but it'll make much more sense on the next slide. But this B form is inactive, but it can be an active form too under a certain circumstance that we're going to talk about right here. So it's kind of nice segue, I guess. Here's again, we're going to talk about the little like mechanism, how glycogen phosphorylase works. So again, this modification, the covalent modification, is catalyzed by these two enzymes. One's called the phosphorylase kinase, which adds the phosphorol group to this enzyme, which is glycogen phosphorylase. And the other enzyme involved for this modification is phosphoprotein phosphatase, or glycogen phosphorylase phosphatase. These are the same thing, and that gets rid of this phosphorol group of this enzyme, and that works basically. Adding the phosphate will make this phosphorylase in the active form, but also we talked about the allosteric here, right? So it has the allosteric effector site, and the allosteric effector for this enzyme, which is glycogen phosphorylase, is AMP. So whenever there's AMP bound to the allosteric site, then even if there's no phosphorol groups that are attached there, it can still be active. Make sense? So it works in two modes. One is for the covalent modification, and the other is for the allosteric. So this one basically shows the same thing. In the similar way, when ATP is introduced into this active form, it goes back to the inactive form. And that kind of makes sense because it is all about the generating energies, right, in the metabolic pathway. So if you think that there are enough energy sources there, which are the ATP, you don't want to make any excess ATP there. So working as a sort of a homeostasis here, or the equilibrium, where it's not going to generate more of the ATP, so or the process the glucose here. So what it's going to do, glycogen here. So what it's going to do is that it'll become the inactive form. When there's a little ATP around, but lots of AMP, which is a used form of ATP, so it already lost the energy there. Then it can use the as a cue for the it has to work on to generate more energies. And similar thing applies to the this phosphorylation too. And it all depends on the ratio of these A form and the B form of glycogen phosphorylase to determine the which way the glycogen process should go, breaking down or forming it up again. Question. Yes, so here, it inactivates it. So AMP will activate the glycogen phosphorylase. And here it says ATP, or glucose 6-phosphate, or glucose, or caffeine, will inactivate this phosphorylase. Oh, yeah. In the same way too, for this one too, as well as here too. So here or here too, I mean, it says there. But regardless it's with the phosphorol group or not, since it still have the allosteric effector site open, so it'll be affected by those modulators. All right. Again, this is all about this allosteric by allosteric effector site, as well as covalent bonding too for the phosphorylation. And rest of 3 were the basically reversible binding of other proteins, irreversible covalent modifications, and changes of the localization of the enzymes. And you can actually see all these happening in serine proteases that we already talked about, which has a critical role in digestive system. And 3 examples that we talked about were trypsin, chymotrypsin, and elastase. And trypsin comes in the inactive form as a sort of zymogen, if you can recall. And this will be bound to the processing protein first to become an active tyrosine. And this tyrosine will work with the chymotrypsin and elastase to further activate these two enzymes. So it's gonna work in it's gonna be trypsin will be work as an other protein, binding protein for these two enzymes. And then they're going to work together basically because they have a different specificity for their targets, but working in the similar places. And they are generated, made in the pancreas, but will be secreted in the small intestine. So they change their localization for their enzymatic activities too. And again, here are the third, fourth, and fifth method to change or modulate the enzyme's activity. Again, reversible binding of other proteins. Another example is pancreatic trypsin inhibitor here. And this inhibitor tightly binds to the active site and prevents the trypsin that is prematurely activated. So it has to be activated in the small intestine, not in the pancreas, right? So it's working through this binding of other protein. And irreversible covalent modification will be the cutting down these zymogens, the precursor of the enzymes, which do not have um, enzymatic activity yet. And that happens a lot in this through the cascade of the proteolytic activations. When the hormones released upon the some kind of like mechanical force or the detection on your stomach, then basically it releases enteropeptidase that will bind to the trypsinogen, which is a zymogen for trypsin, will activate the trypsin by proteolytic activity. So it'll cut down the unnecessary parts, revealing the active site of trypsin. And this trypsin can further activate the zymogens for the chymotrypsin and protease, elastase, eventually will lead to the activation of these two for further digestion. And also changes of localization, as we talked about, there's a different places in that the zymogens produced and the actual enzymes are working. And these zymogens are going to be trapped in granules once it's produced in the pancreas, especially in the exocrine cells, and then will be released upon the cell activation again. It can be the hormonic activation or the some kind of other like mechanical detection or the force on your stomach or the small intestine, where it'll release these vesicles to the out to the plasma membrane, will release those enzymes to the duodenum. And so far we talked about the metabolism, especially just lastly focusing on the digestive system. But metabolism actually refers to the process of breaking down or making the molecules inside the cell. And these red highlighted organelles are the ones that where metabolic processes happens, like lysosomes breaking down the many proteins, and as well as lipids, chloroplasts making the energies there, cell walls, vacuoles, and so on. And importantly, mitochondria is just a factory for the energy, like bioenergy. And to better understand how this metabolic process happens, we need to understand the chemistry of these biomolecules, how they are working and what's the property of those molecules. And most of the biomolecules are composed of the carbon, and this carbon is so important as we talked about, throughout this course, because it has the four valencies. It's the highest valencies, so it can make the up to 4 covalent bonding with the other atoms. And also, so it takes up the over the half of dry weight of the cell as well as living organism. And it can form the single bond with the hydrogen atom, which is important because it's highly abundant in the aqueous solution or the aqueous environment. And also it can make the single or double bond with oxygen, which is important for the generating energies and processing the um, metabolism, basically, in the living system. And also it can make these bonds with the nitrogen too. And it can make the bonding with the other carbon too. So, and that can be single, double, or triple bonding, uh, which all gives the different variety of properties in terms of different properties for the carbon usages, making different molecules there. And also all the enzymes are designed in a way that the working specifically on these bondings, carbon to carbon or carbon to oxygen, to break it down depending on the different types of a bonding, like single or double bonding. And these are the functional groups that we already talked about, and these are especially important and widely used for the bio molecules. And we already used a lot of many of them here. For example, hydroxyl groups in many amino acid side chains, carbonyl groups like aldehyde and ketones, carboxyl groups, amino group, and amido group, which is like important also because of the shift base, and disulfide bonding, thioester bonding, ester bonding, sulfur group, and so on, basically here, phosphor group and stuff like that. These are all important. As well as a phosphodiester bond is important where, it connects the nucleotides too. And all these determine the chemical properties of those, different molecules that are going through the metabolism. And again, enzymes are attacking especially on these functional groups to break it down. An example of these like molecule that containing functional groups, especially multiple ones, is this coenzyme A that contains acetyl group, pantothenic acid, as well as diphosphate and so on, methyl group, imidazole, and so on. So all the molecules have all those like widely used common functional groups there. Okay, and this is kind of the important process for the metabolism, where the carbons can be oxidated, so oxidized, and carbons have a different oxidation state, which are 5 states here, where this methane is in the least oxidized form, and the carbon dioxide is the most oxidized form. And this matches with this sort of molecular writing here, can alkane, alcohol, aldehyde, and so on. And this process of oxidation is basically losing the electrons. And you can take it as the gaining the oxygen too. It's either gaining the oxygen, losing the electrons from the compounds that has to be processed during the oxidation. And oxidation of carbon, you can take it as these two cases where the carbon-hydrogen bonding changes to carbon-oxygen bonding. And carbon-hydrogen bonding, when you look at the carbon-hydrogen bonding, the carbon is relatively more electronegative compared to hydrogen. So these carbon will basically owns or will have the electrons more closer compared to the hydrogen. So it'll be less oxidized form, as opposed to it becomes it is oxidized to become the hydrogen oxygen bonds, in which case oxygen is much more electronegative. So it'll actually pull the oxygens and retract it more like deprive it from the carbon. So carbon will be like less with the less electrons. So you can take it as the loss of electrons. Again, when the oxygen or the more electronegative atoms are bound to carbon, it'll consider as the oxidation or oxidized form where the carbon loses the electrons. And generally, oxidation reactions combines the energy release. And most living cells obtain the energy that is needed for the sustaining the life, basically all the cellular work by oxidizing metabolic fuels like the whatever you get from your diet. And this catabolic pathway, which is energy yielding process of metabolism, is the oxidative process. So you're gonna go through the multiple oxidations to gain the energies on each steps of oxidation, and ended up resulting the carbon dioxide here. And again, the oxidation is about losing the electrons from these energy sources, and these electrons should be transferred to the other molecules, especially ones that accepts the electrons like oxygens, directly or indirectly through the electron carriers. Again, oxidation process is really important for the metabolism. And of course, this reaction should, combine with again, this is about losing the electron. So some other molecules should gain the electron too, right? So this combined process or the reaction is called oxidation reduction reaction or redox reaction in short, and that involves the electron transfer. So when you look at the molecule here in the diagram, molecule A, this one already contains two electrons, but it loses electrons. So losing electron meaning that it is oxidized. So it goes through the oxidation, and there is another coupled reaction here where the molecule B accepts these electrons. So gaining the electron, meaning that the molecule B is reduced through the reduction here. And this oxidation and reduction process or the reactions are always combined, like I told you. And it is all about the whether losing or gaining the electrons. And since in the oxidation reaction, this A molecule loses electron and oxidized, but it actually induces the gaining of the electrons for the B molecule, which is reduced, right? So it induces the reduction of the B molecule. So the A molecule will be called reducing agent. Although A molecule is oxidized, it'll be called reducing agent, as opposed to this, B molecule that is reduced will oxidize the A molecule. So it'll be called oxidizing reagent, oxidizing agent. Okay, and in actual biological oxidations, just it's not about just losing electrons, but it also loses two hydrogen cations in the form of two hydrogen atoms. So it's the same as two hydrogen atoms, right? Which is pretty neutral, but it's in the end losing the two electrons. And it's commonly called dehydrogenation because you're losing hydrogen atoms from your initial molecule. And the enzymes that are involved in this process is called dehydrogenase or dehydrogenases. And every oxidation again should be combined or accompanied by the reduction process too, where it accepts those electrons that were taken from the other molecule that goes through the oxidation. And those electrons will be accepted by um, here the I didn't actually say that, but the electron transfers such as NAD and the FAD. So here's an example of redox reaction where lactate is oxidized to pyruvate. And here the lactate loses two electrons as well as two hydrogen cations on this reaction to become pyruvate, and it's losing these two hydrogen atoms basically from this C2 of the lactate, and these free electrons are accepted by the NAD plus, which is an electron transfer. So that means it accepts the NAD plus. So NAD plus is reduced to NADH. So you can take it as here, from the bottom, lactate. It becomes pyruvate by the lactate dehydrogenase. And NAD plus works as an oxidizing agent to accept the two electrons to become NADH and H plus. And the reverse reaction would be basically, flipped here. So pyruvate will be reduced to become lactate, meaning that it gains electrons, and NADH loses electrons, so it's going to be oxidized to NAD plus. And again, these electron transfers are really critical for the oxidation reduction reactions. And among different electron carriers or transfers, these two coenzymes are widely used, which is NAD plus, nicotinamide adenine dinucleotide, and FAD, flavin adenine dinucleotide. And this produces these two electrons as well as two hydrogen cations after oxidation. But this hydrogen cations can be soluble because it's an ionized form of the water molecule, as opposed to electrons cannot just move by itself, and so it requires carriers. So that's why these NAD or FAD electron carriers are necessary for redox reactions. And I believe this is the last slide. There are two subclasses of enzymes that are involved for oxidation reduction reactions, where the molecular oxygen works as a participant. The first one is oxidases, which catalyzes oxidation reactions, but the molecular oxygen serves as an electron acceptor, but it is not involved in the actual process here. So when you look at it, this glucose oxidase is an example of these oxidases, where this glucose is converted to the gluconolactone here, and it ended up making hydrogen peroxide, and here oxygen was not incorporated or combined to make the final product, but it only accepts the electrons in the form of these hydrogen hydrogen atoms. The second class is oxygenases, which catalyze the reactions of the oxygen atom, but oxygen atoms will be incorporated into the product in this case. And it's also the induces the oxidation, but by adding the oxygen here. So here's an example where there's a phenyl ring here, and using this toluene deoxygenase, it'll add the two hydroxyl groups. So basically adding the oxygen molecules there, or inducing the oxidation of this molecule. All right, so that's it. And next we will keep discussing the other metabolic pathways.